Rejuvenate Biomed, and she is one of the very rare species in our industry who have credible biotechnology and biopharmaceutical background and are running a startup which is clinical stage and targeting an age-related disease and the fundamental process of aging. Please take the floor.
Thank you so much for a very nice introduction. It's my pleasure to be here, and I know everybody's soaked with information. We're on Friday, it's a marvelous conference, but I also know that a lot of information has been coming to you guys. So please bear with me and allow me to introduce you to the world of Rejuvenate Biomed and how we would like to contribute to the field. So we are founded in 2017, so we're still a quite young company. I'm the founder and I have the privilege to also be the CEO, but I'm very driven, of course, because of my passion. It's a little bit a different way of starting a company. I'm a scientist from background, so I had to learn a lot. We're located in Belgium, very close to the border with the Netherlands and Germany. We develop combination drugs which are IP protected, and it's all based on platforms. So we're a very data-driven organization. Of course, it's our aim, just as all of you, to delay the onset of age-related diseases. There's not much need of an introduction here. We're talking here to an audience which is really focused on it. We all know that these diseases of aging are a major healthcare challenge, but we also know that getting old is actually a privilege. It brings a lot of knowledge and wisdom to the world, so let's make sure that we can continue to grab that knowledge and bring it to the next level. However, what we also see is that the urgency to tackle the real driver of these age-related diseases, being aging itself, somehow gets lost. There are always other things which are more important, which are more acute, that people want to focus on. On top of that is also a challenging field, so we should also be able to call a cat. The science behind it, you all are contributing to it. It's very multifactorial. It's not just one target like how we used to think about drug development when we talk about specific diseases. We also know that these age-related diseases have a long incubation period, so it's also not that easy to tackle it from the clinical perspective. And on top of that, if this is not enough, we have the regulatory challenge. However, we're so motivated, we will be able to succeed and we will be able to deal with all these elements as long as we all work together. It was always very difficult for us to explain to people not in the aging field what it was so much that happened to the elderly. Unfortunately, something dramatic had to happen, and it was COVID, where everybody now started to realize that yes, this older population is more vulnerable. And we need to do something with it. We really need to help them to make sure that we can give them a better life. And it's not enough to react in an acute setting. We need to do something to prevent that this acute event happens again. It also teaches us that there is not always time to look at completely new molecules that have to go through a long safety route that need to be looked at for efficacy in multiple settings. So people did start to look at compounds which were already out there and checked if they could be combined and then in this acute setting already make a difference. We also know that it was not only the elderly that suffered from everything that happened because of COVID, but also the younger generation. There was a mental impact, but also a functional impact. So you do see that what happens to elderly is definitely also relevant for the younger generation. So what did we learn from that? We learned a couple of things. We learned that yes, you can have a chronic setting that can lead to an acute problem. So it's important to focus on a condition where you can see acute problems, but you know that the underlying problem is a chronic one. We need to be able to provide solutions to help not only the elderly, but also that can contribute already to the younger population as well, to think as broad as we potentially can and not too narrow. Having the established safety at hand is definitely a benefit. It makes that through the drug development, where you have to go from phase 1 to phase 2, phase 3, and so on, you have to focus on safety. In phase 1, it's the first element that you look at, is your primary endpoint. regulation is as it is, and we can complain about it, and we can nag about it, that it's too long, and it doesn't go fast enough, and they should change, and they should do this, and they should do that, or we can try to do something ourselves. So there are a lot of options there, but no way to hide, because it will come all down to ourselves. So we took all these learnings, and actually this is something that we already apply since 2017, and we'll talk a little bit on our way of working. So what we do is we do actually start with compounds that have been out there already for quite a long time. We check if they are safe enough to go to a chronic setting, because a product that is used for an acute setting might have a very different safety profile than something that you would like to use for a long term. So we take that into account. We are agnostic for the indication for which these compounds are on the market. We only check for safety. And the next steps, when we have that information available, we check how these compounds would be able to impact the longevity genes, but also the longevity pathways. Then we check if we can find compounds that will be able to have an impact on different pathways, creating a synergistic effect. When we have that information, we of course check if we can have IP around it, because as people have told you already yesterday, no IP, no company. It's as simple as that. It's a hard world out here. Then when we have this information, we move to our animal models. So we have a platform on C. elegans. We check there if this, what we predicted, also holds. And these are not little people, obviously, but you did hear a lot already about the strength of C. elegans. You can pull out a lot of information. We're also setting up, and that's very preliminary, we're evaluating the killifish as an additional model. And then we move into mice. We always take 2 mouse models. We take one genetic model that represents the indication that we will be studying, and then we take normal old mice. Of course, these are very long trials that we're doing, but we think it's important to have these 2 approaches next to each other. When we have that information, we move into the clinic. So we do a phase 1. As I mentioned, safety is already known. We do have a new combination. We have admit-tox information available, so we can predict that the combination is safe. However, you still need to show it. So you need to do the phase 1. And I will show you later on how we did the phase 1, where the primary endpoint indeed is safety, but which allows us to already explore a lot of different elements as well. After that, we'll do a phase 2, where we can do the proof of concept in the clinic. Subsequently, we will look for partners to do phase 3s, to bring products on the markets. That's not our thing. We like more that translational part of what everybody is doing here together with you to bring it to the next level. I know I have a lot to talk about, and I can talk for ages, but Martin promised me to give me a signal when I have 3 minutes, so I might not get to the end, but please bear with me, and I'll try to get as far as possible. So there are a lot of in silico platforms, and everybody needs it a little bit, uses it a little bit different. The sources are a bit different, how you evaluate them, how you normalize the data. All this is a little bit different in different platforms, but it all comes down to the same thing, that you take the data, you look how you can use them, you get new knowledge out of it, and these insights are then being used for predictions. We use these insights to predict how they can impact the longevity pathways, but also how this specific combination, knowing now that they can impact this specific pathway, for which disease would they then be the best solution. So we include that part as well. On C. elegans, we of course want to do a lot. So only working on the manual on plate was not fast enough for us. So we have explored possibilities for other systems. So we're currently working with a microfluidic system, because in our case, which might be different for everyone else, we need repeated dosing, which made it life a little bit difficult. We just opened up our own lab at the university campus in Hasselt, and we're very proud that we now are up and running. As I mentioned, we do have a regulatory challenge. We try to go as broad as possible with the first indication, but it also needs to be data-driven. The data have pushed us in the direction of muscle. Within the muscle, you have sarcopenia, which is sharing a lot of the age-related dysregulation pathways, so we decided to move forward with sarcopenia. As most of you know, it's skeletal muscle failure. You see that people are losing their physical ability, it's muscle mass, and most important, muscle quality that is deteriorating. The good thing about this disease is that it does have an ICD-10, so it means you do have a regulatory path, it has endpoints, it has also a diagnostic set that is being internationally used. It also has another element, which I also mentioned before. There's an acute setting and there's a chronic setting. The chronic sarcopenia comes with age. Nothing needs to happen, we only need to age. The other one is an acute setting, which is coming from muscle disuse. And that's something that can happen to any of us. And I know I'm a little bit older than most of you, but also this younger generation will have moments that they, because they went skiing or because of other reasons, have a period of bed rest or immobilization where you notice that you see your muscle shrinking away, a little bit scary when you come out of a cast. Having that ability of that acute setting creates also the opportunity to do a lot of evaluations faster, but you then can translate. That this is a growing area is quite clear, no words need to be spent on that. But having that opportunity, because aging is really where we want to go. That's the flag up there on the mountain, that's the flag we want to reach. But we need to do that within the constraints of the reality. So that's why we're taking that stepwise approach to create early on already a kind of return of investment, a contribution to the community, but still with the focus of having aging in the background. So in all the work we do, even if we focus on muscle, we add in elements looking at aging elements in general. We do have IP in place. It was a challenge, we did it. First element has been granted. We do have the data in place, so we know now much better than at the beginning what the mechanisms is, because we started, of course, the other way around. We started by looking at the functional outcome and then determined what the mechanisms are. I'm not going into detail here because Avi will be bringing out a publication really soon, so I'm not going to steal her thunder. I will show you a couple of data here. This is the mouse data in OPA1 knockout, and OPA1 knockout is creating a problem specifically in the muscle. We knocked it out, so it's a mitochondrial dysfunction system that you create. And we looked at the three elements which are important for sarcopenia, but also in general aging, obviously. Muscle mass, yes, we could see that RGX-01 did improve the muscle mass, where the individual compounds had no effect. Physical performance, we put them on a treadmill, we let them run. There again, the combination was not just outperforming, but we did not see any effect of the individual compounds, which was for us really something that we did not expect. The owner of this model, Professor Marco Sandri, had never seen such an impact of an intervention before. So we were all very happy and very curious, but we wanted to understand what was happening. As I mentioned, muscle strength is most likely the most important element in sarcopenia, more than pure mass. And so what we did is we anesthetized the mice, we opened up the leg, we simulated the nerve, and we measured the muscle contraction. And here we could see again that RGX-01 was doing better than the control and the individual compounds. We now wanted to know, is this because of the fact that we have more muscle, that we have a bigger strength? So we normalized by dividing by the mass to see if the muscle itself was functioning better. And yes, we could see that after normalization, RGX-01 was still outperforming the individual compounds. As I mentioned, we also then moved to old mice. We looked what's happening there. And again, here we looked at physical performance. Again, RGX-01 was doing better than the control. Muscle strength. Again, here we could see an improvement. Unfortunately, due to COVID, we were not able to do all the muscle strength measurements as we had planned it out for. We had to be creative. Well, actually, our collaborators had to be creative to see how they could still have these measurements included, knowing that they were only allowed into the lab for 3 hours or maximum 4 hours a day. So there was not only the scientific challenge, but also the practical challenge to live in a world that just looked a little bit different at that moment in time. We also looked at the neuromuscular junction because with sarcopenia we know that, that there is neuronal health, muscle health, inflammation, but definitely also the neuromuscular junction, the crosstalk between the neurons and the muscle. So in green you will see the presynaptic side of the nerve. In red, you will see the postsynaptic side of the muscle. These two colors need to be aligned. If you don't have the neuromuscular junction aligned on both sides, there's not going to be any transfer of information. We saw that with RGX-01, we could really keep more neuromuscular junctions alive and active. We also want to know, of course, a little bit about regeneration. How did that look like? And we have some data on that as well. So on the, I'm now starting the other way around, sorry for that. So the muscle satellite cells, we wanted to see, are there enough satellite cells available to also still create new muscle if needed? And we saw that with treatment of RGX-01, there were more of these available than without. I already talked about the function of the muscle itself. We did not really understand what was going on because we saw all these phenotypic outcomes, but we were not sure what's happening at the molecular level or at the histological level. So the TEM data that you see here on the left side, is it left side? Yes, it's left side. You see at the upper panel, you see on the first picture that indeed, not 3 minutes yet, huh? Okay, so I'm gonna move forward. What you see here is that even visually, you can see that it functions better. The sarcomeres are aligned, and also the mitochondria are swollen and not active anymore. I quickly want to say something about the phase 1 study. The phase 1 study is currently ongoing. It's a randomized, placebo-controlled, double-blind study, which we're performing in people which are at the age of 65 to 75. We have Silke here today as well, our CMO, so if you have questions, she can definitely answer them. What we do here is, as I said, we first want to do it because we need to, from a regulatory perspective. We will be looking at safety. We have to hold with. We're not too concerned about that. Up till now, we did not see any negative effects, so that's already good. And we will immobilize them. So we immobilize them from the leg up here up to the foot for 2 weeks. They're staying within the CRO or within the assigned location. And then we have a whole bunch of measurements. Purely for sarcopenia, we don't need to do all these things. But for aging, we want to include a couple of things. So what we basically do is we create a human model like we create in animals. Here, of course, there is in animals as well an ethical component. Here, there's an ethical component to it as well. So what we do here is, before they go into cast, we take a muscle biopsy. They have a lot of exercises that need to be done. We measure them all. We have MRI, DEXA, ultrasound, because these three are still a little bit in the field, like unclear which one is the best, so we're learning for the next study. But we're also including a lot of exploratory endpoints, so we will be taking a lot of blood to look at biomarkers that we have predefined, but also markers that we can still do later on, based on the knowledge that you guys are all creating. We, of course, want to learn more about the biomarkers and see what happens. Phase 2 will be shortly starting as well. We had a clinical advisory board last week, Friday, so we're now compiling the information to really fine-tune that phase 2 study. This is where we are at right now, and these are the wonderful people that are giving guidance to the company. We're, of course, not doing that alone with the management, but we also have a wonderful team behind us internally as well as externally. So with that being said, I would like to thank all our collaborators. Without you guys, we cannot do it. Without all of you here in the audience contributing in any way that you do, we are really, really thankful to all of you. And we will do our utmost best to also contribute to the field in any way we can. That was all I had to say today.
Thank you. Thank you. Thank you so much. So we are, of course, over time, but we need to ask questions because you are, as a matter of fact, a wonderful sponsor of the conference. Yeah. So, yeah.
I hope it's not
and
for
thank
that
you for that, and thank you for a really great talk. What I'm missing here is your preclinical models are not reflecting your clinical study. you didn't have immobilization model in your, you know,
Correct.
so that's one question. The second question, you really didn't use an age model for your studies. What I'm getting at is sarcopenia is not what you're looking at in this study. I'm sure you know that. And I actually think that sarcopenia is a really tough issue.
Absolutely.
You know, we want to prevent sarcopenia at the end, right? So
Yep.
how are you dealing with those two things?
Yeah, it's a little bit this schizophrenic way that we have to think about these things, like one way of bringing it to an indication where you can really show that there is an indication for which it can work. And then in the meantime, really focusing on that aging component, which sometimes make that you are like in this in-between world. Also with the preclinical data, we focus much more on aging than specifically on sarcopenia, but the data were good enough to convince the Fach that we could move forward already in the clinic to look at muscle components. So it is a challenge, and it's like almost on a weekly basis that we have to think about, is this strategy still working for the two worlds? One world which is not defined, and one world which is defined. Yeah, it's not an easy one. And especially towards investors, right? Now, fortunately, we have investors that understand what we want to accomplish in aging. Before, when we started, nobody knew, and they all looked at me like, huh, aging? What are you going to do in aging? There was no investment world available at all. Now, finally, it's coming, but we now still stick to the two approaches, the short-term return of investment and then the aging component. The thing that we work on most is making sure that we have the safety at hand, because unsafety can damage.
I saw on your slides that you have a very nice improvement of PEX7 cells that are, to my knowledge, muscle progenitors. I'm curious about actually differentiation to myoblast and myosatellites. So did you check MyoD and MIF5 markers? Are the improvements there?
Yeah, so a lot of that work is ongoing and part has been finalized. So I will leave that up to Evie Merke, who will be presenting all the scientific backgrounds, and she will also put it in the full framework. But more to come. Yeah.
All right, thank you very much. But one question. So those are a combination of FDA-approved drugs already?
Yes.
Okay, so
And
you basically took
EMA-approved.
two FDA-approved drugs, combined them, and are testing them.
Yes.
Great, got it. Thank you. So